Okay. So today I'm going to talk about a new FDA approved treatment for major depressive disorder and the study, one of the main studies that the FDA approval was based off of. So the new treatment is called transcranial direct current stimulation, or tDCS. Now, tDCS is not new. It's been around for decades and it's been studied for a long time. There's lots of studies on tDCS. However, most of the data so far has been pretty mixed. There was a meta analysis showing that there was a potentially small effect in major depressive disorder. If you looked at individual patient data and kind of pooled it from all of the sham controlled studies, they saw like a 30% response rate, 20% remission rate. So that's, pretty bad. And just a lot of heterogeneity in the data. You'd see some trials that showed it seemed to be effective, other trials showing a negative results. So it really never went anywhere for a long time because of the mixed data. So I was pretty surprised to see that just recently the FDA approved a specific TCS device by a company called Flow Neuroscience. I think it's a UK company based on a study called the Empower trial that was published in Nature Medicine, which is a very good journal. So I guess let me back up and tell you what tDCS is if you're not familiar with it. So as the name suggests, you're using some kind of electrical stimulation. And it's basically you put two electrodes on the head. So one electrode goes on the left side of the head, left side of the forehead. The right electrode goes on the right side of the forehead, and you pass a small electrical current through the electrodes. The current is to some degree passing through the brain, primarily those prefrontal cortex areas. So let me just contrast this with electroconvulsive therapy, or ECT, because on the surface, they're very similar technologically. In ECT, you're also putting electrodes on the head and passing a current through it. However, in ECT, we're using a pulsed alternating current. So you're sending electrical current in both directions, so to speak, in pulses. And you're using a pretty high amplitude, high amperage current. Typically, we're using 800 milliamps. Okay. And that's enough to induce a seizure, which is the whole point of ECT. And so it's done under general anesthesia. It's quite a big ordeal to do ECT because of all that. For tDCS, you're using a very small current, only usually on the range of 1 to 2 milliamps. Okay, so that's less than 100 times the current of ECT. It's not enough to induce a seizure. For most people, it's barely perceptible. You might feel a little tingling sensation on the skin where the electrodes are. If you don't have good electrical contact, some side effects could include mild skin burns or irritation of the skin area. But in general, if you have good electrical contact, you use electrodes that are high quality and have good conductivity, it's fairly noticeable current. Again, you might have some tingling sensation. Now, in ECT, we think of the seizure as being the main source of the clinical effect. And so we spend a lot of time trying to diagnose issues when we're not getting a good seizure. So it's interesting that tDCS seems to possibly be effective with such a small current, not inducing a seizure. Okay. And I should say in tDCS, you are passing just a direct current. So that's why it's called transcranial direct current stimulation. So it's just a small constant current between these two electrodes. There is a related technology called transcranial alternating current stimulation. And as the name suggests, that uses an alternating current instead of direct current. And some people argue that might be more effective because it basically opens up a new parameter space for adjusting the treatment. But anyways, I'm going to focus on tDCS. So how does tDCS work, or how do we think it works? It's kind of hard to say. The physics of it are both simple and complicated. By applying these electrodes and passing a small constant current between them, you generate an electric field across the brain. And this electric field can influence in a very small way the neurons. It can either make the neurons a little bit more likely to fire an action potential or a little less likely to fire an action potential by slightly depolarizing the axon initial segment area of the neuron or slightly hyperpolarizing that area, which is the part of the neuron that initiates the action potential. And this really depends on the orientation of the neuron relative to the electric field. So if you've ever seen like a picture of electric field lines between two electrodes, they're kind of these curled lines that emanate from the anode, which is the positive electrode, and kind of sweep over to the negative electrode, which is the cathode, in a sort of arc shape. And so if the neuron, or more specifically, if the main axon direction is oriented along the electric field, the main electric field axis, then you will get general a slight depolarization of the neuron. If the neuron is oriented perpendicular to the electric field will basically not do anything to it. And if the neuron is oriented parallel, but in the opposite direction, it can hyperpolarize, very slightly hyperpolarize the neuron because the electric field in this case is so weak in tDCS. So the overall effect is very heterogeneous. You're going to have some neurons that are slightly biased toward activity and some neurons that are slightly biased towards suppression. And the net effect is just going to depend on the average orientation of the neurons through which this electric field is acting. And I think that might go a long way toward explaining why there's so much heterogeneity in the data that we see. Sometimes it works, sometimes it doesn't work. Again, the effects can be very different just depending on the orientation of the electric field, the orientation of the brain tissue in relation to the electric field, so on and so forth. And also just that with such a small electric current, the different effects of resistance are going to matter a lot more compared to something like ECT. So with all that said, let's talk about the study that led to the FDA approving this device. And again, the company that makes it is called Flow Neuroscience. The specific device that was studied is called the FL100 by Flow Neuroscience. And in this study, this was a randomized controlled trial where they had an active tDCS group that was using their device. And their device has the electrodes in a bifrontal placement montage. So both electrodes, again, are targeting the kind of prefrontal cortex areas using a 2 milliamp current, direct current. Their study was 10 weeks. For the first 3 weeks, patients were doing 30-minute sessions for 5 days a week. And then for the last 7 weeks, they were doing 3 sessions a week, also for 30 minutes. And one of the interesting things about this study is that it was a totally home-based study. So I guess they just sent all the study participants the FL100 tDCS device. They gave them instructions. They had virtual appointments for study check-ins and all of that. So it was a totally home-based protocol. And I guess let's just go into the results. So their primary endpoint was the average change in the Hamilton Depression Rating Scale score, the AMD. And if you compute an effect size from the change in HAMD from active versus sham, you get an effect size of about 0.37. And just to put that into perspective, that would be considered a fairly small, maybe moderate at best effect size. And effect sizes are just a way to standardize a treatment effect so that you can be able to compare across different treatments in a way that's not specifically using one type of measurement. Because different studies might use the AMD, might use the MADRS, might use the quiz scale. So if you want to be able to compare how effective a treatment is when different studies are using different rating scales, then you really want to be paying attention to the effect size. So SSRIs have an effect size of about zero point 0.3 to zero point 0.4, somewhere in that range for major depressive disorder. So this would suggest it's in a similar range. And the interesting thing is that they had a significantly higher response and remission rate. Their response rate was about 58%, and their remission rate was about 45%. So that is surprisingly high for such a small effect size. And my guess is that they are having a lot of heterogeneity in the data. So you're having some people that have a very big response, that are going into remission. And then you have a lot of people who are barely responding or not responding at all. So when you average it all out, the average is not very impressive. But if you look at the remission or response rates, you do see a lot of people reaching remission, as opposed to something else where everybody just gets slightly better. So hardly anybody goes into remission, but the average still is about the same. One potential problem with this study is the blinding. So 77% of the people in the active group were able to correctly guess that they had the active treatment and not the sham. And I should say the sham treatment was they had the exact same device, and it gave them a little tingle at the beginning and the end, but it was not active in the middle, basically. So I wonder how much of the unblinding could account for the effects, because if you know you're not in the treatment group because you're not feeling the tingling, especially if you maybe have some exposure to tDCS in the past, then that might be a little bit of a nocebo effect where you get disappointed and it might suppress your response, right? So it's pretty harmless treatment, so maybe worth giving a try. It's about $500 out of pocket to buy the device, and they also offer a rental option for about $80 a month. So yeah, something worth considering. So that's all I wanted to say about this interesting new research on transcranial direct current stimulation and this new FDA approved option for major depressive disorder. Hope that was helpful.